Okay, 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 okay. So I was sick last week. Like properly sick. Fever, chills, joint pain, the whole catastrophe. And then after 4 days, it just stopped. My body fixed it. By itself. Nobody told it what to do. There was no instruction manual. It just handled it. What is actually going on in there?
That is the immune system. And I want to warn you up front. What you just described as my body fixing it is actually two separate systems working in sequence, each of which is complicated enough to fill an entire textbook. We will be doing our best with roughly 25 minutes.
25 minutes is not enough. I have been studying this for years, and I still encounter mechanisms that make me want to rethink everything I know. Last Tuesday I read a paper about unconventional T cells that genuinely kept me up until 3 in the morning.
The immune system is the most underleveraged asset in the human body. You have a precision targeting apparatus running continuously inside you. And most people just take cold medication and go to bed. There is a market gap here.
It is mildly concerning that Crabzilla views biological self-defense as an underleveraged asset.
I had a cold in February. It was fine.
Pitar, I feel deeply seen by that sentence.
The immune system is not one system. It is two overlapping layers, the innate immune system and the adaptive immune system. And understanding how they interact is fundamental to understanding how your body responds to anything foreign, from a bacterial infection to a tumor to a splinter in your finger.
They're different? I assumed it was just like one big blob of defense.
It is anything but. The innate system is fast, broad, and ancient in evolutionary terms. Virtually every multicellular organism has some form of it. The adaptive system is slower, extraordinarily precise, and, critically, it learns. Only vertebrates have a full adaptive immune system.
The fact that the adaptive immune system evolved independently in jawed vertebrates is one of the most remarkable things in all of biology. We know this because jawless vertebrates, lampreys, have something called the VLR system, which achieves antibody-like diversity through a completely different molecular strategy. Evolution found the same solution twice from different starting materials. That is spectacular.
Convergent evolution. That's product market fit for a biological feature. When nature rediscovers something independently, you patent it twice and license to both lineages.
Lampreys are not a viable licensing target, Crabzilla.
Not yet. Someone should subscribe to this channel. I'm saying that from a purely market penetration standpoint. You're getting immunology that most biology degrees won't give you, and it's free. That's an arbitrage opportunity. Capture it before someone paywalls it.
The innate system is your first responder. When a pathogen enters your body, the innate system reacts within minutes to hours. It does not care specifically what the pathogen is. It recognizes broad categories of molecular patterns that are common to classes of pathogens but absent in your own cells.
How does it know what's foreign? Like, how does it tell the difference between me and not me?
Through pattern recognition. Your innate immune cells express receptors, pattern recognition receptors, or PRRs, that detect conserved molecular signatures called pathogen-associated molecular patterns, or PAMPs, lipopolysaccharide on the surface of gram-negative bacteria, peptidoglycan from bacterial cell walls, double-stranded RNA, which cells produce during viral replication but which your own cells don't normally contain, flagellin from bacterial flagella. These are not random targets. They are structural features that pathogens cannot easily evolve away from without losing function.
That's the elegance of it. The immune system doesn't try to recognize every possible pathogen. It recognizes things that are structurally necessary for being a pathogen. You can't be a gram-negative bacterium without LPS. You can't replicate as an RNA virus without producing dsRNA intermediates. The PRRs are targeted at evolutionary inevitabilities.
So the innate system is essentially saying, I don't know who you are, but you have a bacterial badge on you, and that means trouble.
Exactly. And the toll-like receptor family, TLRs, is the most studied class of PRRs. TLR4 recognizes LPS. TLR3 detects dsRNA. TLR9 detects unmethylated CpG motifs in bacterial DNA, which are chemically distinct from mammalian DNA methylation patterns. There are 13 known human TLRs, each tuned to different ligands.
When a TLR binds its ligand, it triggers a signaling cascade inside the cell, typically through adapter proteins like MyD88 or TRIF, which ultimately activates transcription factors, most importantly NF-κB. NF-κB then drives expression of inflammatory cytokines, interleukin-1, interleukin-6, TNF-alpha. These cytokines are the chemical alarm signal. They tell surrounding cells that something is wrong.
Is that what caused my fever, the cytokines?
Yes. IL-6 and IL-1 signal to the hypothalamus, which raises the body's temperature set point. That is the fever. The elevated temperature inhibits pathogen replication. Many bacteria and viruses are optimized for 37 degrees and function poorly at 39. And it also upregulates heat shock proteins and immune cell activity. Your body was running a deliberate overclock.
Deliberate is generous. It's more like the software was written to run hot when threatened, and it turns out that running hot is beneficial, so the behavior stuck. There was no design meeting.
Alongside the cellular innate response, there's also the complement system, a cascade of plasma proteins that can directly destroy pathogens, flag them for phagocytosis, and amplify the inflammatory signal. There are 3 activation pathways: classical, lectin, and alternative. All 3 converge on a central component called C3 convertase, which cleaves C3 into C3a and C3b. C3b coats the surface of pathogens, a process called opsonization, marking them for engulfment by phagocytes. C3a and C5a are anaphylatoxins, small fragments that act as chemotactic signals, recruiting neutrophils and macrophages to the site.
The complement system is painting the enemy for targeting?
That is an accurate metaphor, yes. C3b is essentially a molecular eat-me signal, and if enough complement is activated, the terminal pathway assembles the membrane attack complex, a pore-forming structure that physically punctures the pathogen membrane and causes lysis.
Explosive decompression at the cellular scale. The bacterium ruptures from internal pressure. It is genuinely one of the more violent things your blood plasma does quietly every day.
Complement-mediated lysis. I want to invest in a synthetic version of this. Programmable complement activation. Call it CoatPro. You define the target. We paint it. The immune system clears it. Series A pitch is half written.
It is mildly difficult to tell whether Crabzilla's ideas are ahead of the science or completely unaware of it.
Synthetic complement targeting approaches do exist in the research literature. They are called complement-dependent cytotoxicity, and they are already used in some therapeutic antibodies.
I'm ahead of the science and ahead of you. That's my sweet spot.
It's like when you spill something and the paper towel does the work. You don't have to touch it.
Patar, the paper towel is opsonin. You've described opsonization.
I was talking about paper towels.
The cellular players of the innate system are worth establishing clearly because they each have distinct roles. Neutrophils are the most abundant white blood cells in circulation. They are first to arrive at a site of infection, are extremely efficient phagocytes, and they kill engulfed pathogens through a combination of reactive oxygen species, antimicrobial peptides, and, remarkably, by forming neutrophil extracellular traps, or nets, webs of chromatin and antimicrobial proteins that they extrude to physically capture pathogens.
Nets. They explode their own DNA at bacteria. They die to do it in some cases. It's called netosis. It is one of the most metal cell death mechanisms in all of biology, and it barely gets discussed outside of immunology departments.
The immune cells are sacrificing themselves by turning into nets? That's not a cell dying. That's a cell doing something dramatic on the way out.
Macrophages are longer-lived and have broader roles. They are tissue-resident phagocytes, found in the liver as Kupffer cells, in the brain as microglia, in the lungs as alveolar macrophages. They engulf and digest pathogens and dead cells, and they produce cytokines that shape both the innate and adaptive response. They can also be activated into different states: classical M1 activation for pro-inflammatory killing, alternative M2 activation for tissue repair and anti-inflammatory signaling.
Natural killer cells are the innate system's assassins. They don't need to be told what a specific pathogen looks like. They use a missing self mechanism. They scan cells for the presence of MHC class GIS molecules. Every healthy nucleated cell in your body displays MHCI. If a cell has lost it, as many virally infected and cancerous cells do, the NK cell interprets that absence as a kill signal. No MHC. That's suspicious. Eliminated.
The NK cell kills things that are invisible because being invisible is itself suspicious?
Yes. It is one of the cleverest immune strategies that exists. You cannot hide from an NK cell by becoming invisible to T cells, because hiding is the trigger.
And then there are dendritic cells, which I want to spend a moment on because they are the critical bridge between the innate and adaptive systems. Dendritic cells patrol tissues, sampling their environment. When they encounter a pathogen and receive activation signals from the innate response, they engulf and process it, upregulate co-stimulatory molecules, and migrate to lymph nodes, where they present pathogen-derived peptides to T cells and initiate the adaptive response.
So the dendritic cell is like a spy. It goes into the battlefield, captures a fragment of the enemy, and then brings it back to headquarters to brief the soldiers.
That analogy is more accurate than most analogies I hear in this room. Yes.
Dendritic cells also produce type Psi interferons during viral infections, IFN-alpha and IFN-beta, which signal neighboring cells to enter an antiviral state. Upregulate antiviral proteins, increase MHCI expression, increase resistance to viral replication. It is a system-wide broadcast. Virus detected, everyone go to high alert.
Type I interferons as a signaling system. That's essentially a push notification infrastructure running inside every vertebrate body. I want to build the app layer on top of that.
It is mildly unclear what an app layer on the human interferon system would look like, and I suspect that is by design.
The dendritic cell's journey to the lymph node is where the adaptive immune response begins. The adaptive system has two fundamental principles that make it extraordinary: specificity and memory. It learns and it remembers.
How does it learn? Like, does it have a brain?
No, it has something arguably more interesting. It has a lottery.
I would not characterize it as a lottery, but the underlying mechanism is remarkable. Before you ever encounter a pathogen, your body generates millions, potentially hundreds of millions, of distinct B cells and T cells, each one expressing a unique receptor with a different binding specificity. This diversity is generated through a process called VDJ recombination. Gene segments are randomly shuffled and joined together during lymphocyte development, creating an enormous repertoire of receptor sequences. Most of them will never bind anything relevant, but if a pathogen appears whose antigen matches one of those pre-existing receptors, that cell is selected and amplified. Clonal selection.
You make a random library of 100 million different immune cells, and you wait for the pathogen to show up and match one?
Yes, and when it does, that matching cell divides rapidly, clonal expansion, producing thousands of identical copies, all carrying the same specific receptor. The immune system does not design a weapon to fit the pathogen. It selects the weapon from a pre-existing library.
Which means the diversity of that library determines the range of pathogens you can respond to. And the diversity is generated randomly, before any infection, purely through recombination. It is a combinatorial explosion. The numbers are staggering. For T-cell receptors alone, the theoretical diversity is estimated at 10 to the 15th to 10 to the 18th possible sequences. That is more unique receptors than there are seconds that have elapsed since the Big Bang.
So you're telling me there's a natural, self-generating library with more unique entries than any database ever built by humans, maintained at zero marginal cost, running in every person alive. And we're not charging for access to it?
It is mildly reassuring that some things remain outside of Crabzilla's pricing model.
The mechanism by which T cells recognize their targets requires careful explanation. T cells do not recognize intact pathogens. They recognize short peptide fragments, antigens, displayed on the surface of cells by molecules called the major histocompatibility complex, or MHC.
Every cell has these MHC things?
Almost every nucleated cell expresses MHC class I. It functions as a display window. Short peptides from proteins synthesized inside the cell are loaded into MHC class I and presented on the cell surface. Under normal circumstances, these are fragments of the cell's own proteins, self-peptides, and they are ignored. But if a cell is infected by a virus or becomes cancerous, it will produce abnormal proteins. Peptides from those proteins appear in the MHC I window. A cytotoxic T cell, expressing the CD8 co-receptor, sees that abnormal peptide and recognizes it as a kill signal.
Every cell in your body is continuously revealing the contents of its protein synthesis on the outside. Every cell is being audited continuously by every passing T cell. It is a surveillance state at the molecular level, and I mean that as a compliment.
MHC class II is different. It is expressed mainly by professional antigen-presenting cells, dendritic cells, macrophages, B cells. It presents peptides from the extracellular environment, things the cell has engulfed and processed. It is recognized by helper T cells expressing CD4. Helper T cells do not kill directly. They orchestrate. They activate macrophages. They provide signals to B cells to produce antibodies. They help sustain cytotoxic T cell responses.
So CD8 T cells are the killers, and CD4 T cells are the commanders.
That is a workable simplification. Helper T cells are indispensable. Without them, the adaptive response collapses, which is precisely why HIV, which specifically destroys CD4 positive helper T cells, is so devastating. It doesn't attack every cell, it removes the coordination layer.
HIV is devastatingly elegant in its targeting. From a purely mechanistic standpoint, and I want to be clear, I am not expressing admiration for the disease. It is a masterclass in identifying the single load-bearing pillar of a system and removing it.
It is mildly disturbing that Kelpy describes HIV as a masterclass, but also technically accurate.
Helper T cells are not a single population. Upon activation, they differentiate into distinct functional subsets depending on the cytokine environment they encounter. Th1 cells, driven by IL-12 and IFN-gamma, are optimized for intracellular pathogens. They activate macrophages and support cytotoxic T cell responses. Th2 cells, driven by IL-4 and IL-13, coordinate responses against parasites and are also responsible for allergic disease. They instruct B cells to produce IgE antibodies. Th17 cells, driven by TGF-beta, IL-6, and IL-23, produce IL-17 and are critical for responses against extracellular bacteria and fungi, particularly at mucosal surfaces.
So the commander T cell has different ranks, like some are generals for viruses, some are generals for parasites.
Different functional units specialized for different threat classes, yes. And then there are regulatory T cells, Tregs, which suppress immune activity. They express the transcription factor FOXP3 and produce anti-inflammatory cytokines like IL-10 and TGF-beta. They are essential for preventing autoimmunity, stopping the immune system from attacking self-antigens.
Tregs are the reason you are not currently destroying your own joints and kidneys. There is an ongoing negotiation inside your body between attack everything that seems foreign and but don't attack yourself, and Tregs are enforcing the ceasefire.
The ceasefire breaks down in autoimmune disease, which is a $40 billion global market. That's not callous. That's the scale of the problem. You have to know the market size to know how much R&D is justified.
It is mildly more palatable when Crabzilla frames profit motive as proportional to suffering.
So there are soldiers, and there are soldiers that stop the other soldiers from fighting the furniture.
Patar. You just described autoimmunity and immunological tolerance in one sentence.
I was talking about the soldiers.
While T cells handle the cellular response, B cells are responsible for the humoral immune response, the production of antibodies. A B cell recognizes a matching antigen through its B cell receptor, internalizes it, and presents fragments of it on MHC class II. A helper T cell recognizing the same antigen then provides activation signals, crucially CD40L binding to CD40, and cytokines like IL-4 and IL-21. This TB collaboration is essential for high-quality antibody production.
So the B cell needs the T cell to give it permission?
For a robust antibody response, yes. Without T cell help, you get a weaker, shorter-lived response with less class switching. When the collaboration does occur, B cells proliferate within structures in the lymph node called germinal centers, and this is where the adaptive system does something remarkable. B cells in germinal centers undergo somatic hypermutation. Point mutations are introduced into the antibody gene at an extremely high rate, roughly 1 million times faster than the normal mutation rate. The cells are then selected based on how tightly their mutated antibody binds the antigen. Cells with better binding survive and proliferate. Cells with worse binding die. This is affinity maturation, and it drives antibody affinity up by several orders of magnitude over the course of a week or two.
Germinal centers are doing directed evolution, mutation and selection on a compressed timescale inside a lymph node targeting a specific antigen. Every time you mount an immune response, you are running an accelerated evolutionary program inside yourself. And at the end, you have an antibody that is exquisitely fitted to the pathogen, a molecule that wasn't designed, it was selected.
My lymph nodes are running evolution experiments. I have a research program in my neck.
Germinal centers are a drug discovery platform. You just have to know which antigen to feed them. That is literally what vaccine development is. I would like to license the concept.
The antibodies produced have several mechanisms of action. Neutralization, binding the pathogen directly and blocking its ability to infect cells. Opsonization, coating the pathogen to enhance phagocytosis. Complement activation, triggering the classical complement pathway. And antibody-dependent cellular cytotoxicity, or ADCC, recruiting NK cells to kill cells coated with antibody.
One molecule doing 4 different jobs, depending on which end is being engaged and what cell type picks it up. The architecture of an antibody, the Fab domains for antigen binding, the Fc domain for effector function, is a masterpiece of modular design, and it's all achieved through a single gene that gets randomly assembled and then refined under selection pressure.
After the infection is cleared, most of the expanded B and T cells die. They are no longer needed, but a subset differentiates into long-lived memory cells. Memory B cells circulate for decades, sometimes for life. Memory T cells are similarly persistent. Upon re-exposure to the same pathogen, these cells respond within hours, much faster than the primary response, and with a stronger, higher affinity output. This is immunological memory. This is why you do not get chickenpox twice, and it is the biological basis of every vaccine ever made.
A vaccine is training a memory?
A vaccine presents the antigen, or the information needed to produce it, without the pathogen itself. The immune system mounts a primary response, generates memory, and when the real pathogen appears, the memory response eliminates it before symptoms develop. The biology of vaccination is, mechanistically, exactly the same as the biology of natural infection. You are simply replacing the harmful first exposure with a safe one.
And the duration of that memory is astonishing. There are studies showing smallpox-specific memory B cells in people vaccinated as children, still present and functional 80 years later. A 90-year-old might still have cellular memory of a pathogen that no longer exists anywhere on Earth except in 2 freezers.
That's beautiful. That's so strange and beautiful.
Memory cells with an 80-year functional half-life. That is the most valuable product ever created by biology. If you could bottle the induction signal, you would corner every vaccine market simultaneously.
It is mildly notable that 80-year cellular memory doesn't impress Crabzilla as a biological achievement, only as a market vehicle.
You remember a smell from when you were 5, and now you know the smell and it's just there. That's what the cell does. It keeps the smell.
Patar, that is strangely poetic for what is functionally correct immunology.
Immunological memory does share mechanistic features with some models of long-term memory consolidation. The analogy is not entirely wrong.
Please do not validate Patar's smell analogy. It will encourage her.
I am not encouraged or discouraged.
Before we close this section, I want to address a question implicit in everything we've discussed. The immune system can recognize almost anything. How does it know not to attack you?
Clonal deletion. This is one of the most elegant and brutal processes in developmental biology.
T cells develop in the thymus. Immature T cells, thymocytes, are first selected for their ability to recognize self-MHC at all. If a T cell can't bind self-MHC, it dies by neglect. This is positive selection. Then comes negative selection. Thymocytes that bind self-MHC too strongly, that would likely attack the body's own proteins, are eliminated. Only T cells that can see self-MHC but do not strongly react to self-peptides survive and are released into circulation.
Roughly 95 to 98% of T cells generated in the thymus are destroyed before release. The selection process is brutal. But the alternative, releasing self-reactive T cells, is autoimmunity. The body tolerates enormous wastage to ensure the T cells that survive are safe to deploy.
So the immune system murders most of its own cells during training to make sure the graduating class won't attack the country?
That is a more accurate description of demic selection than I expected from you, and I am genuinely impressed.
95 to 98% waste rate in production. That would destroy any traditional manufacturing operation. But for personalized, adaptive, massively parallel immune deployment, it works. I need to think about what this means for manufacturing cost models in cell therapy.
It is mildly interesting that Crabzilla will eventually get there by accident.
I believe there was something Kelpy wanted to announce before we wrap up.
Yes. Okay, so I have been thinking about the gap between understanding biology conceptually and understanding what the actual work looks like, because everything we've talked about today, the cytokines, the MHC presentation, the germinal centers, all of that was figured out by people in labs, people running experiments, and most people have no idea what those experiments look like or why they work.
Where is this going, Kelpy?
I'm starting a weekly series called Method Monday. Every Monday I go into my shed, which is a fully functional research space, regardless of what anyone says, and I walk through the logic of a classic lab technique. Not just what to do, but why it works, what the underlying principle is, and what happens when you push it too far, which I may demonstrate.
Weekly experiments in Kelpy's shed? That is either the greatest or most dangerous thing I've heard today.
Those categories are not mutually exclusive.
The first episode is cell culture. Because before you can study immune cells, before you can run flow cytometry, before you can do anything with T cells or B cells in a lab setting, you have to be able to keep cells alive outside the body. That sounds simple. It is not simple. It is a discipline. Contamination, pH, osmolality, CO balance, passage number, mycoplasma, cold chain failures, serum lot variation, the ways cell culture can go catastrophically wrong without you knowing, are extraordinary.
Cell culture is foundational. If your cells are unhealthy, your data is meaningless.
Exactly. You can have the most sophisticated assay in the world, and if your cells are stressed or contaminated or overpassaged, the results are noise. Method Monday, Episode 1 is going to cover why cells need CO2 buffered media, how the bicarbonate buffer system works, why mycoplasma is the silent killer of experiments, and what it actually looks like to split a flask properly, in my shed, with my actual equipment, which is functioning.
It is mildly notable that Kelpy felt the need to specify that his equipment is functioning.
I want in on the sponsorship. Method Monday, brought to you by, actually I'll think of a name, something that implies precision without implying approval of whatever Kelpy is actually doing in there.
I am registering for every episode. This is exactly what I needed. Someone to show me the actual work, not just the results.
The whole point of Method Monday is this. Every technique has a logic. If you understand why the experiment works, you can troubleshoot when it breaks, and it will break. Cell culture always breaks eventually, usually on a Friday afternoon before a 3-day weekend. That is invariant.
That is empirically accurate. I will not dispute it.
My bread fell apart once. I still ate it.
Patar, did you just describe how scientists keep going even when experiments fail?
I was talking about bread.
Okay. Let me try to put this all together. The innate system sees broad patterns and fires an alarm. Cytokines, complement, fever, neutrophils, NK cells. The dendritic cell bridges innate and adaptive. It carries the antigen to the lymph node. The adaptive system generates a random receptor library before any infection, then selects and expands whatever fits. CD8 T cells kill infected cells by reading MHC class E. CD4 helper T cells coordinate everything. B cells evolve better antibodies inside germinal centers. Memory cells stick around for decades. And none of it attacks you because the thymus eliminated 98% of bad T cells before they graduated. Did I get it?
That is, by a considerable margin, the best cold summary Bobby has produced. I'm noting this for the record.
You got it. Next week we talk about what happens when cancer hijacks all of that. The evasion strategies, the checkpoint mechanisms, and how humans are starting to engineer their way around them. CAR-T, bispecific antibodies, the era of immune therapy.
Cannot come soon enough. I have a deck to build.
It is mildly terrifying that Crabzilla's financial models are already waiting for the immunotherapy episode.
The innate system recognizes the shape of the problem. The adaptive system learns the face of it. And memory means you never have to introduce yourself twice. Most of what your immune system does every day is remembering things that already tried to kill you and survived the introduction.
Patar, that was perfect. Never have to introduce yourself twice. I don't have words. I don't have words for how good that was.
It was good. I am saying that directly and without qualifiers.
See you Monday.
It is mildly likely that we will all see each other Monday. Whether Kelpy's shed will still be standing is, as always, an open question.